Yeah, so we, as you are aware, like we were recently funded by USDA FFAR, and that is a project concerning developing reduced immunogenicity peanut lines. And these lines are to the benefit of the people who are sensitive to peanuts, because peanut allergies are actually about 6 million individuals are suffering from peanut allergy in the United States, and that include the children and adults as well, and it affects the children more than it affects the grown-ups. Um, and at the moment there is no remedy for peanut allergy, so one has to either just quit eating on peanuts completely, and as you are aware, like I mean, the peanut dust can cause allergy. So I mean, a very small quantity of the peanut can actually have a detrimental effect on a person who is sensitive. So that's why like we need to develop some sort of remedies that can actually help the people who are sensitive, and especially like reduce the fear among the parents of these children who are suffering from these peanut allergies, because of these allergies the schools cannot serve the peanut butter and jelly sandwiches over there, which is favorite of students or the young kids. And also like in the airlines, as you are aware, like I mean, the peanuts cannot be served because of this problem. So we can bring the peanuts back, like if we will have some sort of remedy. So that's the target. So there will be two things that we will do, like one, we will be actually looking at the natural germplasm of peanuts, so diverse peanut line collections, that the US Department of Agriculture has a very large collection of peanut lines. And then also like we have access to an international collection from ICRISET, which is an international institution which has like its headquarters in India, but they have their things stocked at USDA. So we have access to both of these collections. We also have access to some of these other lines from South Asia that we will screen for their protein profiles. We have some preliminary idea because we have, before getting this grant proposal, we already run like some of these electrophoretically, so you can actually separate the proteins based on their size and charge, and then you can tell like, okay, if one of the lines is lacking or accumulating these proteins in reduced quantity. So we will screen this large germplasm by different means, which is electrophoresis, chromatography, mass spectrometry, and then also enzyme linked immunosorbent assay. So that's a immune test. You will test these two for the quantity and quality of these proteins. So we will develop a profile of these lines, and then we'll make genetic crosses among the lines that were actually accumulating these proteins that cause allergy, or we call them immunogenic proteins, and then develop some of the lines which will be reduced in the amount of these proteins. And then the other part of this project will be to gene edit these genes that produce these allergens in the peanut grain, and then edit them out, like so they will not produce and accumulate these toxic proteins in the grain. So these are the two parts of it. And then the third component of the project that we will do is like with our partners, they will screen these the lines for their proteins on the human cell lines collected from the individuals who are sensitive to it, and that component will be done in University of North Carolina. So we have a partnership with University of North Carolina, and then we have the North Carolina State University and USDA ARS germplasm collection in Griffin, Georgia. So it's like kind of a 3-state union, which is really important because these are the 3 primary states in the southeast that produce a whole lot of peanuts for the whole of the United States and also to export to other countries. So for this study, we will be largely using the electrophoretic units to run the vertical page gels, and we will be also using like the spectrophotometer that will be kind of doing like the colorimetric assay to quantify how much of these proteins we have, and that will be through the enzyme linked immunosorbent assay. At Clemson main campus, we have like this laboratory, like which is a common use facility called Mule, and that is for the analysis, the chromatographic analysis and mass spectrometry analysis of these proteins. So we have like a whole suite of equipment available down here at PD, and in conjunction with the main campus facilities that we have access to. So we should be able to produce like a detailed protein profile of these lines. I think like I wish to say like the major thing is the partnerships that we have. So we have been partnering with like or have partnership with the people who have developed the oral immunotherapy for peanut allergies that has been approved in 2020. It's called Pelforsia. The issues with that thing is it's fairly expensive, and the moment you stop eating like that or going for this immunotherapy, you will be go back to the ground zero. So you cannot quit eating it, and the thing is fairly expensive. At times it could cost like 10k, and so we were trying to have something similar to it but a more natural solution, or a solution that is more affordable to the large community like down here or in the other countries like which cannot even afford like that kind of treatment. And those people we have partnered with, so they will analyze this material further. So we have these good partnerships right with the people who have invented the oral immunotherapy.